I'm very happy to be with you, being with you in quotes. I have spoken, I believe, 2 or 3 times to your group up there in the Twin Cities, and I've always enjoyed being with you. Sorry we can't be with you together live. This is the next best thing, whether you're looking at us on this live Zoom meeting presentation, or you'll be viewing this later on the YouTube channel. I have a variety of presentations, and this one is something I've put together recently because when I do talk about human ancestors, it's mostly about the bones, but this question always comes up: human and chimp DNA. I think most of you are familiar with my background. I am a retired engineer / engineering manager. I worked for a Fortune 100 company for 38 years. Became a Christian while studying engineering at the University of Illinois. But I always had questions, even back then, like what about the Lucy fossil? What about dinosaurs? Like my buddy Rex behind me. What about carbon-14? And so I've talked about some of these things. And I have learned a lot in the last, 38 years. It's become a hobby looking at creation science. How much of the Bible can I really trust? Is it, what about those first few chapters? Are they allegory or are they literal history? Well, they're written as literal history, but we all have questions. So the question has come up, did we come from apes? I mean, Darwin proposed that, and it's been in our culture ever since. Unfortunately, Darwin was a racist, and he really believed that darker skinned people were closer to the apes, less human. That's a very unfortunate idea that floats out there, even today, and I hope tonight we can dispel some of that. Human chimp DNA is the main topic for tonight, but what I want to do is just do a little recap about the bones. Now, this is a picture I took in the Chicago Museum of Natural History. They have a separate exhibit called The Evolving Planet. I was there, took a lot of pictures. This picture was taken 10 years ago. And not everybody realized it. This was put up by the evolutionists in charge of that exhibit. I did not draw that red line through there. They did. And the note in that sign, this is an untouched sign or picture of a sign. Hominid species did not all evolve in a single direct progression from apes. And that is right. That whole picture is falling apart. We do have a collapse of the traditional understanding of ape to man evolution. Of human evolution that you'll find all over the place, says Dr. Wood in Who Are We? A New Scientist. This was already 18 years ago. On the left, the picture, there's an ape. On the right, a man. And between the two is a succession of figures that become ever more like humans. Our progress from ape to human looks so smooth, so tidy. It's such a beguiling image that even the experts are loath to let it go. And that's very true. They don't want to let go of it, but they have to, because as Dr. Wood says, it is an illusion. Now, that doesn't mean they're giving up on evolutionary theory. No, not at all. But I just want to drive home the point, even in the premier science magazine Nature in the year 2000, we have a quote: the once popular fresco showing a single file of marching hominids becoming ever more vertical, tall, and hairless now appears to be fiction. Wow, for Nature to call it fiction. And no wonder. In my other talk, when I concentrate on human evolution from apes and go through all the bones, because that's been where a lot of the story has been ever since Darwin, beginning with Piltdown man and then Cro-Magnon man, Heidelberg man, Neanderthals are in there, Egaster and Erectus. Well, those last 5 that I mentioned, you could see in white on the screen, those bones have already now been determined to be fully human. Yeah, there's a few people trying to hang on that Neanderthals or some of these others aren't quite fully human, but they are. They have, all these bones have the same amount of variation as we get into the details that people among us do. All of us have some variation, right? Slightly different skull shapes, but we're all human. Slightly different rib shapes, but we're all human. And there's very detailed things as we learn more and more about these bones that these are all human, and then there's a few others. These are the more popular ones. And then Rudolfensis, habilis, or handyman afriensis, which is the Lucy fossil, and her cousins, which we have found. Good thing we found more bones of Australopithecus afriensis, because they all show themselves as we get things like the foot fossils, which Lucy didn't have, to show that they are true chimpanzee or chimpanzee-like creatures. Sediba, Artie, or Arthropithecus ramidus, is shown to be an ape, probably an orangutan. And then Ida, which was very famous in 2009, it just looks like a very squished lemur. So really, the bones don't show us much about evolution, and the more we look into their details and find more matching pieces from excavations, the whole story has really fallen apart. Finally, also in Science magazine, more recently in 2015, we have a quote: "The field is in crisis. It's a messy, tangled bush. There is no fossil trail to man." Quote, "If we want to be objective, we shall almost certainly have to scrap the iconic list of names in which hominin fossil species have historically been trapped." I like that word. They trapped them into that picture. But we have to rethink the genera and species. Now, obviously, they're not giving up on evolution. I'm not saying that. What I'm saying is the bones do not show any kind of ape-like creature to human progression through evolution. The bones have not shown that as we learned more and more about them, despite all the claims and beautiful pictures that we've been shown. Well, back in 2007, Newsweek magazine had a feature article that said the evolution revolution. And this was kind of a recognition that the bones were not doing what they wanted. In fact, you can find pictures of what's called a tangled bush. It's not that straight line progression. And then there's a lot, even today, missing links. So in this article, they admit this, that fossils never resolved when the lineage is split. Yeah, they still have faith in evolution, that we came from some type of ape-like ancestor. But here, when they admitted this, they also had something else in their pocket. And that was the new study of DNA and mapping of the genome and all the stuff that was going on in the 2000s. So here's that quote: "Fossils never resolved when the lineage is split, but DNA might." That was their hope, DNA might. So they started looking more and more at DNA. And of course, this was about the time that we were getting the first versions of the chimp DNA sequences. And so in the same article, practically the next sentence, it says, "Human DNA and chimp DNA differ by no more than 1.2%. How many of you heard that? 98.8% the same. And really, when you think about it, that's about as far apart as they can be and still have evolution occur in about 6 million years. So they're hanging on to that 1.2%. And I'm going to show you how pervasive that has become. When I just talk about the bones, that's always a very popular question from the audience at the end. You know, hey, what about that number? What about human and chimp DNA being 98.8% the same? Doesn't that prove human evolution, that we came from some ape-like ancestor? Well, I'm going to try and finish that story up a little more today with some more DNA information. So I've given you as much as I'm going to on the hominid fossils in today's presentation, but the DNA similarity issue is there. And there are 3 main points that we'll look at tonight. One, what about that 98.8 similarity? It's often been cited I still hear it today, and you probably do too. But there's another issue behind that one, and that's called a fusion site on human chromosome number 2. I'll get into the details, but really quickly we'll look at that, because all apes and monkeys, all of the ape-like creatures have 24 pairs of chromosomes. Humans have 23, and we'll get into that. And, you know, what if this evolution could be? You know, if you've got a few percent, whatever it is, difference, how long would it take to evolve chimp DNA to human DNA? And that is formally called the wait time problem. How much time does it take for natural processes of evolution to evolve from a chimp-like creature's DNA to the human DNA that we know today? I'm going to finish with a little extra on human development and implications, because DNA is such an important role as we develop, not just in our mom's womb, but as we grow as children, and the growth of the human brain especially. We'll look at that. So 98.8% number, that was researched and calculated by scientists who already believed that we are descendants of chimps. And having that preconceived notion, no matter how independent and scientific people try to be, it does influence our results, even as all scientists have to admit at one point or another, unless you've got hard, clear, step-by-step logical progressions, mathematical things that you can point to and other people can reproduce. That's why to be scientific and to do scientific experiments, you have to have that repeatability function, that other people can get the same results. How often do we get the 98.8% number? Well, you'll see it really wasn't that often, and there is a lot of confusion. Part of it already began with the mapping of the chimp genome. The human genome, we said we mapped it on, it was announced that in 2001 we had it pretty much. That was their goal in those days. But I read today even that there's still about 5% missing. Now we fill in the blanks with things that we think might be there or just repeated nucleotides. But even today it's not 100% chromosome by chromosome, exact, here is the genome. We don't have that. Not 100% even today. But let's say we do have it, and we were really close, no problem with that in 2001. But that's the other issue, though, is how do you compare it to something like the chimp genome? Well, they had working on pieces of it. Certain chromosomes had been mapped in the early 2000s for the chimpanzees, but it was not even completely coded or issued until 2005, and I'll get into that, and I'll show you how that led to the 98.8% number. But when you get into those details, and they didn't always announce this, so if you got into the details, you could find it. They used the human genome as a scaffold. This is where that belief, that bias comes in. If you firmly believe, well, evolution is true, there's so much proof, we came from apes. Darwin said so, 160 some years ago, whatever. Well, they must be similar DNA. Therefore, they thought it would be fair to use the human genome as a scaffold or an outline. And so as they map pieces of the chimp genome, and really this is not an easy thing to do. I mean, we're talking at the super microscopic level here, and you can't just open it up and read it like a book. So as they map pieces of it, they fitted it to the human genome scaffold, which kind of influences any comparison you're going to make. And of course, as best as we could, scientists couldn't help but get some human DNA onto their chimp samples. So there is some, especially in those days, human DNA contamination in this first sequence of the chimp DNA. And today it's still not 100% complete either, much less than the human. So those are things to keep in mind as we look at some of these comparisons and studies. especially as, lay people, we're trusting the scientists. And then we get the all excited media out there, and they publish the headlines, and a lot of people just look at the headline. Here's a quote from Dr. Jeffrey Tompkins, who has his PhD in genetics from Clemson. The chimpanzee genome was literally put together to resemble the human genome. Well, we evolved from apes, so they must be similar. So let's just start with that as an assumption. Dr. Tompkins says this little known fact was accomplished by taking small snippets of DNA produced after sequencing and lining them up on the human genome. The human genome guided the researchers throughout the chimp genome assembly process. That was especially true in the years from when they started the chimp genome in the early 2000s until they started issuing some more of these up until 2013, and I think we've got even better versions today. So this is one of the major articles that started this number, this 98.8. I think I actually heard about it before, but let's go with this. 2005, Nature, the premier science magazine, had an article where they announced the sequencing of the chimpanzee. Now, I already gave you some of the issues that were there already in 2005. But one of their headlines was, look, we've got the initial sequence of the chimpanzee genome and a comparison with the human genome. Ooh. And here's their finding number one. Single nucleotide substitutions occurred at a mean rate of 1.23% between copies of human and chimp DNA, with 1.06 or less corresponding to fixed divergence between the species. So when they look at just the DNA number, the big one that jumped out at everybody, 1.2% difference between copies of human and chimp. That's it. That's the 98.8% difference. So in the next few years, we had articles, not just the Newsweek magazine that I already showed you, but in 2006, Time magazine had a big article. It said how we became human. And I talk about that picture they use on the cover. But down at the bottom, right on the cover and in the article, it says chimps and humans share almost 99% of their DNA. And the new discoveries reveal how we can be so alike and yet so different. Now, I mentioned that this is not an easy thing to do. It's just difficult. And so different people using slightly different methods did come up with different numbers. And that should have been a clue to the problem with some of the numbers that everybody went with. Because in the same year, National Geographic had an article that said humans and chimps are 96% the same gene study finds. So someone else came up with a 4% difference. But this is the one that everybody went with. It's a very premier magazine, and that's what they said had to be. Well, as late as 2012, everybody still talks about that. And of course, people have realized you really can't be too far apart if we really evolved from them, even in 6 million years. I mean, the mutation rates would not allow us to be that far apart, much more apart genetically. So in Science magazine, the online version, here's a quote. Ever since researchers sequenced the chimp genome in 2005, they have known that humans share about 99% of our DNA with chimpanzees, making them our closest living relatives. So this number, 2012, is really being pushed around everywhere. I mean, you just do a Google search, how similar chimp DNA, human DNA is, and you'll find all kinds of answers, and they're always zeroing in on this 98.8. In fact, just earlier this year, there was a debate between two Christians, but Michael Behe, who's known for his intelligent design book, Darwin's Black Box, and more recently, his book, Darwin Devolves, which is a great read. Michael Behe debated Josh Swamidass. Behe does not believe in evolution. Josh Swamidass does, and he's a very articulate young man that really pushes evolution. And so they had this debate. I think that was down in Texas. That was just a few months ago. And in the review of the debate, Dr. Swamidas, in his presentation, he did sketch out a few areas where they agree on, but then Swamidas, as part of his defense of evolution, said that humans and chimps are 98% similar genetically. There's that same number again. You know, he didn't bother the 98.2, but you know, we know what he means. So it's very pervasive. It's very pervasive. Remember, the human chimp DNA, back in, even before they announced it, they thought it had to be similar. So when they studied just parts of it, they were already influenced under that assumption. In a 2003 study, they came up with even a closer number. And what you have to remember is what you had to dig for was that they only looked at 97 genes, 97 protein coding regions of DNA. And then they said, okay, look at, we get the same protein codes in chimps and humans in these 97 protein coding genes. And in those days, they still believed in junk DNA, so they ignored 95% of the genome or more. And they said, wow, look, chimps and humans are even more closely related than previously thought. So there, you can see there's a lot of numbers out there, but they're always in the same range. In these early studies, they only looked at 97 genes, but the current count is that we've got about 20,000 genes that code for proteins and as many non-coding genes. And today we know they're active. They're the controlling genes that do control other genes that control the protein coding genes. And of course, this calculation, a lot of these early calculations ignored 96% of the genome. An area of controversy, something that causes these differences and how they come up with these numbers, is they ignore what's called an indel. An indel is shortened word for insertions and deletions. They only matched where we have sequence of code, you know, AGT, AGT, TC, AG, you know, and so on. And if there's one where there's a C on one side and the chimp and a G on the other, well, then they call that a difference. But then there's big sections of code that kind of skip in one and have extra code in the other before they start lining up again. So besides ignoring 96 of the percent of the genome, they also ignored indels, insertions and deletions. So if you look at this top one, you have a simple substitution that was counted, AGTG, TAC, AGTA, TAC. Okay, there is a difference, everything around it is the same. But this is an insertion. A G is inserted between the two T's here, but there's nothing there. So here it's called a deletion. But everything else is the same. So they ignored the G over here and just said AGT, TAC, AGT, TAC. Oh, look, that little sequence has a 100% match. Of course, you've got to go into the thousands here. But that's an idea of what an indel is. Substitution, yeah, they have to count that. But they didn't count indels. And some indels can be pretty big chunks of code before the same things repeat or start matching up again. Most human chimp differences are nucleotides that have been added on one or the other. And some of these insertions are up to 300 base pairs. That's a lot. That's like putting several paragraphs into a sentence where the rest of the sentence might match on one side or the other. So that because of this indels, which was known to be an issue, but they ignored in the other science study for one thing, in the proceedings of the National Academy of Science in 2003, some of the authors admit the sequence analysis confirms the existence of a high degree of sequence similarity. However, and importantly, this 98.7 sequence identity drops to only 86.7%, taking into account multiple insertions, deletions, or indels, which I think you've figured that out, what they are. And in another study, they only looked at chimp chromosome 22 and human chromosome 21. And in those days, you know, we thought we had pretty good lineup of these two chromosomes, which should be, comparable. And there was only a 1.44% difference here, pretty close to that 98 point something. But even then, they ignored 68,000 insertions and deletions. And again, some of these are pretty big chunks of code. So they did say this: the genomic changes after speciation and biological sequences seem to be more complex than originally hypothesized. Now, part of that is because some indels are a result of a pre-coded selection of our DNA. It's pretty complicated. Like some of these chromosomes kick in our immune system. The way our immune system learns is that it actually does naturally, either by mutation or by some type of artificial intelligence, does perform insertions in human DNA to help us learn to fight off certain diseases and viruses. Another hot topic. But the degree of these is that it can't all be naturally occurring mutations that cause insertions and deletions. There's just too many, and that's why they're starting to get a little nervous about this. And that's why they said, things seem to be more complex than we thought. And it got to the point as we're getting better copies of the chimp genome that seems every few years somebody's publishing an update, that by 2012, a leading primate evolutionist, Dr. Todd Preuss from Emory University, I mean, he is also a strong believer in evolution, but he keeps up on the research. He's involved in human and chimp brain development. And he said this: it's now clear that the genetic differences between humans and chimpanzees are far more extensive than previously thought. Their genomes are not 98 or 99% identical. And he said that and picked up on that from the research in 2012. Too bad Josh Swamidass has still not picked up on that. By the year 2013, there were other studies, including by a creationist who was Dr. Tompkins, but also an evolutionist, a Dr. Bugs from England. They kind of looked at the two different studies and they came up with a 70% similarity. And that was pretty big. I mean, that would rule out evolution all by itself, end of story. But even then they knew there might be more to the story. Plus, in the intervening years, we have learned that some of the algorithms that came from government agencies like the NIH had some bugs in them, and it was kicking out too much data. Plus, we've also had a continual updating of the chimp genome. So we are getting better at this. And we're also learning more about the role of indels and the immune systems, as well as what's going on. So that's what's been going on recently. But finally, in 2018, we've got at least a third generation chimp genome and a continuing updates to the human genome and a better understanding of how to do these, better computer programs, better technology. So Dr. Richard Bugs, who has a PhD in genetics, or primarily plant genetics, he's from Oxford. He is currently at the professor at Queen Mary University in London. And he had this to say on some matchup work that he was doing. And he even talks about it on his own website, how similar are human and chimpanzee genomes. He does not get into the implications. But as he looks at the data and looks at all the studies going on, he came up and said this. He says, guys, the percentage of nucleotides in the human genome that had one-to-one exact matches in the chimpanzee genome was 84.38%. Well, instead of 98.8, this leading genetic evolutionist now says 84.38. So remember that number. That was pretty recently. That seems to be about the best number we have today. Meanwhile, Dr. Jeffrey Tompkins, who has his PhD in genetics also, that's from Clemson. He was in the faculty of the Department of Genetics and Biochemistry at Clemson for a decade. He has published 57 secular research papers in peer-reviewed scientific journals, 7 chapters in scientific books in the areas of genetics. And he was actually the director of Clemson Environmental Genetics Laboratory at Clemson. But he wanted to get more into creation-oriented research, and he took all his skills and knowledge and is now at ICR and a little bit more free to publish how he sees what's coming down the pike in science. And in 2018, he published this, a comparison of chimpanzee sections of DNA to the human. And he goes into all the details, all the methods and algorithms, and which genome he downloaded from, the USC site is a popular one. And he goes through all the details to come up with some of his answer. And he actually, in a totally different approach, came up with an answer very similar to Dr. Buggs over in England. In 2018, Dr. Tompkins said the alignable regions of the chimpanzee sequencing contigs were only 84.4% identical to the respective matches. Well, he left out a digit. Dr. Buggs had 84.38. That's a phenomenal amount of difference when you consider the genome is 3 billion nucleotides long, or base pairs long. He goes on and says, in addition, on average, only about a third of each contig could be aligned using liberal gap extension parameters. In other words, he's giving them as much doubt as he can, making this a conservative number that he gets at the end. And he says the 84.4% nucleotide identity of the alignment is not an indicator of overall genome similarity because it does not include the regions of these sections that are so different that they are not alignable. So even here, even though he comes up with a similar number of 84.4, it could be less. There's still so much more we don't know and because of the complexity and the various issues, it's hard to compare them. But here's the take home from this, where we are today in 2018 data, and we're now in early 2020. And Dr. Tompkins wrote about this at Acts and Facts in 2018. Talked about his study as well as Dr. Buggs. And he said a 15% DNA difference between humans and chimps is a discrepancy that can't be ignored. No more than about a 1% difference is required to make human evolution seem at all plausible. That's the key word, plausible. We'll get into that some more. So at this point, the old 98.8% number is wrong. We've got to remember that small sections of DNA were cherry-picked for comparison in these early published numbers. And they picked the ones that they were already known to be similar. And the first chimp DNA models were based on human genome evolutionary bias shortcuts. Yeah, it takes a long time. It's hard to do this. And since we believe in evolution, they took some pretty biased shortcuts. And that's where the 98.8% number came from. Unfortunately, it's still being used today. No one should use that number anymore. 85% similarity, you can just round it up to that. That's more accurate. It's probably even less similar, but this already rules out evolution. Now, I mentioned something. There is something very unique between human and chimp DNA. Chimps have 24 pairs of chromosomes. Humans have 23. Well, that's a big difference right there. Evolutionists then say, well, oh, wait a minute. If humans have 23, and all chimps, all monkeys, all apes, everybody on that side, they have 24 pairs. Well, the obvious answer is, well, then 2 chimp chromosomes must have fused together into 1 human chromosome at some point in our evolutionary history. That's been proposed, for a long time now, ever since we figured out that they were different numbers. Probably for 20 years, that's been the belief. But people have been researching it. They had no evidence, and then they did have some evidence, and then they finally said, hey, look, human chromosome 2 is pretty long. Chimp chromosomes, and we designated them 2A and 2B, are pretty short. They must have fused together. So they started searching human chromosome 2, and sure enough, there is a believed fusion point. So take a look at this diagram. In the upper left, we've got chimpanzee chromosome 2A and 2B. And at the end, somehow, they fused together at a fusion site. Now, one thing, just a little aside, so you know, at the end of our chromosomes, and chimp chromosomes, all chromosomes, the genome is divided into, in our case, 23 pairs. At the end of the chromosomes is what's called telomeres. And these telomeres are kind of like strong, long strands of genetic code that kind of acts as a signal to the genome when it's reacting and moving around. Hey, this is the end of this chromosome. And especially important when they have cell division and these things divide. The two pairs come apart, and then they got to float around, and then they got to find the other pair when there is a conception going on, cell division in some cases. Either way, that's how they keep track of the ends. And then another point is a centromere. If you remember seeing these pictures, like this one on the right is a pretty, I think, electron microscope picture. There's like a pinch point between the pairs. That's how they know to find each other when they get back together. This half and this half now become the same pair. Let's call it chromosome 2, and this diagram shows you. And the centromeres for different chromosomes have different locations that helps them line up so that the telomeres at the opposite end now fit again, are in the right places, and the centromere now connects where the DNA strands come together to make a single pair. Okay, so those are two very important facts to remember. So when you look at this fusion idea, there's two things to consider. If this is chromosome 2A from the chimp, it's got a centromere. This is chromosome 2B from the chimp, and it also has to have a centromere, otherwise you would not get more little chimps. And all this works. So if this fused, and here's the fusion site, and scientists did think they had identified a fusion site. They actually found it in some type of section of the human chromosome 2 and suggested with the coding that they use, that's where the fusion site is. That's where this happened. So not only does this have to look like a fusion site where the telomeres kind of got mixed up and then banded and then formed into a single human chromosome 2, but you've also got to account for two chimpanzee centromeres. So there is one that they thought was close enough on the human one for one of the chimp centromeres, but the other one has to be now cryptic or unused. It's kind of just a little leftover, unused DNA that used to be a centromere, but now isn't. So there's the fusion site coming from the two ends, the telomeres being lined up. And then here is the cryptic issue. Is there a sign of a cryptic section of a past centromere on the human chromosome 2? So those are the two things to think about. But by the way, there's also 24 million base pairs missing from the chimp, because these are not exactly an exact length when you add them together. There's a big section here missing. So kind of three issues, but we'll look at this first one, the fusion site. Dr. Tompkins, again, PhD, he's done all this study when he was at the Center for Genetics in Clemson. He wrote a paper in 2013 looking in much more detail at the supposed fusion site. And what he found is, and studying this, is that this fusion site is a functional DNA binding domain. And if you read the research paper, you can find it online, or I can send it to you. But this is an expressed gene, and it's in 255 different cell or tissue types. Wait a minute, that can't be a fusion site with just a bunch of leftover telomeres there holding it together. This fact that this is a functional DNA gene where the fusion site's supposed to be totally refutes the claim that it's a fusion site from chromosome 2 of an ancestral end-to-end fusion. It cannot be, because it goes clear across the fusion site. The fusion site's somewhere in the middle there. And if you want to look it up, that's the detailed intron reference point on human chromosome 2. Turns out fully functioning gene. If it's fully functional, it cannot be a fusion site. It's important. We could not have been missing it all this time. And there are 24 million base pairs missing. So that's a big difference between chromosome 2 and 2A and 2B, which they try to match up in some of these studies. And second, that centromere site, there's no sign of it. They've looked at that and looked for it, found a couple of candidates, and have been ruled out because they too are important genes and could not have been a former centromere site. So the whole idea of human chromosome 2 being a fusion of monkey or ape type chromosomes 2A and 2B, that's been refuted in several ways. It cannot be. And yet I still hear people talking about it because they need a story because of the difference between 24 pairs and 23 pairs. The next part of the DNA story I want to mention is, what about the time we're talking about? We're getting an idea of how long, how many, what the mutation rate is for monkeys and chimps and for humans. So how much time would it take to change an ape-like creature into a human? And we're talking about, even if you only go with the 1.5%, we're talking about 75 million letters, millions of new genetic words. I mean, there are major differences between us and chimps. It's not just having 4 arms and 4 legs and a face. It's very different. So with computer modeling these days, this can be studied because there is some problem. Scientists, evolutionary biologists like Michael Lynch are realizing that, we don't really have a good mechanism for evolution to work. Yeah, everybody's taught and we all repeat it. We talk about, the mutations. Most mutations are bad, but natural selection picks the good ones. Well, Dr. Lynch admits a central problem in evolutionary theory concerns the mechanisms by which adaptations requiring multiple mutations emerge in natural populations. Now, that last phrase is important. We need multiple mutations, not just starting to substitute single nucleotides. We've got whole sections of code that got to be quite different to make our bone structure different than Lucy's bone structure, or our muscles and tendons and all the connections. And then there's the brain. There's huge differences in brain and between the two. And I'll get a little bit more into that. So they admit they don't have a good problem, but that's the only story they have. And so a simulation to answer this question was done. And we assumed only a 2.5% difference, or 75 million letters in the genome. How long would it take to make this? Well, the important thing to remember is not just the probability of a mutation happening and then selecting for a mutation that happens to be good. So we're on the third part of this, the DNA question, and the waiting time problem in a model hominid. Now, this was published by Dr. John Sanford, who became a Christian at age 50 and realized all his work in genetics fits the biblical model instead of the evolutionary model. But for 50 years, he believed it. He was an evolutionary professor at Cornell. But he published this in a secular, peer-reviewed scientific journal in December of 2015. And the scientists have already known about what I called the wait problem. And here the headline is, the waiting time problem in a model hominid mutation. And so what they did is try to analyze how long would it take. I mean, we're substituting good mutations in this old genome of the chimp-like creature on its way to becoming human. Well, you need a lot of them, but you got to remember, every mutation that's going to be good not only has to be selected, but it has to become what's called fixed in the model or in the population. You can't just have a single, ape-like creature get this mutation and suddenly, it's everywhere. It's got to be found in the majority of the population, which means in both males and females, and then got to be reproduced and becomes dominant. That's the point of fixation when a mutation becomes the new normal within the population. So he explains what they did here, and the coding that they did in this simulation, they made it open and let evolutionists look at it and say, okay, here's what we think. And they have not found anything wrong with it. What they did is simulated a classic pre-human hominin population of at least 10,000 individuals. And that's normally the story. There were about 10,000 ape-like creatures that suddenly were becoming human. And they gave them a generation time of 20 years, a very strong selection rate. Random point mutations were generated with a starting string. Whenever an instance of a starting string arose, all individuals carrying the string were assigned reproductive advantage. In other words, they put in every possible advantage for the evolutionary story within the parameters of the simulation. And then when natural selection had successfully amplified an instance of the target string to the point of fixation, in other words, hey, this mutation has now become dominant within the population, and then they could tabulate the results. So what was it? Well, they started with only, I think, less than 5%. That means 150 million nucleotides have to be different between the chimp-like and the human. And I need a new substring in the human. Now that percentage times 150 million difference, you're talking about 15 million pieces of code in the genome that have to change. They have to become now dominant to start becoming more human. And this is even with a small number of DNA differences between humans and chimps. So what's the wait time to generate a single DNA? Now they're just talking about a single fixed DNA substring of only 2 to 8 nucleotides. I mean, that's tiny compared to the millions you need to go from chimp to ape. What would that be? Well, here's the results. To establish a string of just 2 nucleotides required an average of 84 million years. Can mutations do that? Well, everybody says it can. We just took it by faith. But now when you start putting some numbers to it, you get ridiculous long wait times. That's why it's called the wait time problem. And then they did it for several runs with different amounts of nucleotides that had to be changed. And to get 5 nucleotides of a good mutation established and fixed in the population, that took an average of 2 billion years. And they say using the most generous, feasible parameter settings, the waiting time was consistently prohibitive. Now remember, the evolutionary guess is that we went from a Lucy-type ape to human in about 3.5 million years. But to do just even minor changes in DNA, you're talking 84 million years to billions. And again, this research paper was published and nobody has found major faults with it or the computer code. It was published in a secular journal 2015. Their conclusion: we show that the waiting time problem is a significant constraint on the macroevolution of the classic hominin population. Routine establishment of specific beneficial strings of 2 or more nucleotides becomes problematic. Oh man, we can't do it. There's just too many changes have to happen, not enough time. So you look at all the fancy scientific journal jargon, and you end up with there's not enough time for random mutations and natural selection to change ape DNA into human DNA. Not even close. So here's a review of the 3 items that I mentioned in the DNA section, the main point of this talk. The 98.8% number was deceptive from the very beginning and just flat wrong. Currently, the 85% is the best guess. That's based on studies by both a secular and a creation-oriented geneticist, but both using the latest genetic methods and information. Second, the supposed human chromosome fusion site has been debunked, genetically impossible. Humans and apes are very different in their DNA, just if you count the chromosomes. And finally, we have a giant wait time problem to change even a little bit of chimp DNA to human DNA. Does not work. So you start putting all this information together and you start realizing between the weight, the fusion problem, the similarities, which are big, humans and chimps cannot share a common ancestor. Not even in a billion years. I think that's pretty significant. Well, how many of you heard of Dr. Stuart Burgess? He's a PhD in mechanical engineering. What does that have to do with this? Well, I think it has a lot to do with early childhood development. Apparently, a lot of our DNA controls not just as we grow in the womb from a fertilized egg, but throughout our early childhood. We are very different from the apes. Dr. Burgess, who is an amazing engineer, he's not just a professor, but he helped with spacecraft design for the European Space Agency. He got a gold medal for his design of a satellite solar array. A whole type of new gearbox was used. He's published 160 scientific papers. He even helped the British Olympic cycling team win 5 championships. Anyway, he designed the new sprocket for a bicycle cycling team in Britain, and the British team won 5 championships and 6 gold medals. 2000, early 2000, he wrote this book, The Design and Origin of Man. And he's got some interesting thing on human development, and I think DNA is what makes us different, even as it controls what we do and how we grow our brain. Humans, as we've been studied them, as we grow from birth, we have like a 225% increase in our brain size. Those of you that are parents, you hold your tiny child at birth, and you realize just how much it grows, and even the brain probably grows the most, at 225% or more from the time of birth. On the other hand, chimps, their brain only grows about 25% to adulthood since the time of their birth. So a quite different story. Additionally, human brain, during our first 2 years, we add 14 million neurons, brain cells, per hour. It's crazy how much we are growing. But even more than that, for every new cell, there's 1,000 new connections. There are amazing connections. And by the end of childhood, the human brain has about 100,000 billion connections that not only help you be you, but to think and process and learn. Human children have an astounding ability to learn and learn at a fast rate. Just on the issue of language, from age 2 on, children learn over 10,000 new words every year, maybe more if they're in a bilingual home. Apes and monkeys have virtually no vocabulary whatsoever. There have been researchers who spent 6 years trying to teach chimps to talk. The best they could get to was like 4 different kinds of grunt. A grunt for being happy, a grunt for being angry or afraid, but no speech. They don't have the wiring for speech. They just don't have it. Not only that, humans can speak at a fast rate for long periods. During this time, it's possible to convey vast amounts of information, ideas, and knowledge. No animal can do that. Got it? The ability to speak and write shows that humans are spiritual beings with design, communicate thoughts and emotions. We can communicate complex ideas. The important use of language is to enable people to find God and to relate to him. God has revealed himself to mankind through the Bible, that is his written word. The ability to speak and write shows that humans are spiritual beings. We communicate not just thoughts and needs, but abstract ideas, emotions, and we can build on them from each other. Dr. Burgess goes on, he says, language and speech enable humans to enjoy relationships with other people, communicating abstract thoughts and ideas. And childhood is the perfect solution for enabling people to develop our human abilities. The length of childhood means that a person can acquire a vast amount of knowledge and skills. Strong family bond skill also are made during childhood, and it's also a wonderful opportunity for a person to develop as a spiritual being. We all know this naturally, I think, especially those of who are Christian parents. I have 3 kids and 6 grandkids. Those early years of teaching love and security in the family are so key, and even secular researchers are realizing that. Of course, Jesus said Let the children alone. Do not hinder them from coming to me, for the kingdom of heaven belongs to such as these. Dr. Burgess makes a lot of other points in his book about how, we do need to learn these things as a child, but including the spiritual things. This is very, very important. Statistics show that most people, if they don't come to Christ by the age of 18, the odds are much against them. It still happens, like Dr. Sanford is one example, but it becomes very difficult. And that's why we need help getting this kind of information out. Because the world teaches this. This is just recent from the Smithsonian National Museum of Natural History. Look at the top. This is what they're teaching your kids. We've got the different types of monkeys lined up, going from lemurs, all the way up to the apes and great apes. And right among the apes, we've got humans. See, we're just connected to all these other types of apes. But we've got a lot of evolutionary leaders out there teaching that if you tell your kids about God and creation, well, that's child abuse. In the UK, they had articles about that saying Christian creation indoctrination is a form of child abuse. Lawrence Krauss even said that. He said he's heard teaching creationism is child abuse, and I think it is. Dawkins has said something similar. Really? What do you think happens when you have generations of people being taught that they're nothing but fancy monkeys? I think the ultimate child abuse is to teach children that they are nothing but fancy monkeys. What kind of results, what kind of society do you get? Not only is that scientifically wrong, it's morally wrong. And that's why we need more of you out there correcting some of the scientific problems that lead to moral decline and decay. We are created in the image of God. We are special. Everything about us is special, from our DNA to our brains. But we also have a spirit, and that's what we need to teach our kids. So what are you and your children going to trust? You're going to trust the Holy Bible, God's Word, which changes not? Or are you going to trust man's word, which is always changing? They never get the story straight. So what we need to do, I'm so happy to be with you today, and I hope you can share more of this. Our group and the Twin City group as well. We're just trying to follow 2 Corinthians 10:5. We are destroying speculations and every lofty thing raised up against the knowledge of God. We need that more in our culture. To drift away from God leads us to a lot of the problems that we're seeing even in our headlines today. Raise the children right. Teach them the right facts. Teach them how to refute some of these numbers. We're not that much like apes.
Hey, could I ask a question? This is Brian. As far as your ministry, Helmut, do you see that the doors are easier to open now as far as getting creation into schools and colleges or other venues? Or is it the same, or is it getting harder?
Well, I think what we're seeing is a divergence from at least what I see. There used to be that it was very difficult to get into a lot of churches.
Right.
But they're starting to realize, hey, the kids we graduated from youth group 10 years ago haven't come back. They don't even go to any church. And as they start looking at that and realizing the statistics are true, 2/3 of kids just walk away. And more they're realizing that, we can reverse those statistics by teaching these kind of scientific facts. Right. And that the statistics are reversed. We can have 2/3 or more of young people who graduate, go on to college, they can earn their degrees, they can do all the wonderful stuff, they're smart, in fact, probably better disciplined, but instead of walking away from the church, they know how to answer the right questions the right way to get the right grades, but they don't have to personally believe it, and therefore they will stay active in a Christian or church ministry. We've actually seen that. So I think we're seeing a little more opening among Christian people, realizing that in the churches and Christian schools. On the other hand, while I meet people that are open because they've never heard anything like this, especially when you can, just ask them a question. Hey, do you remember that old 98.8% number? Do you know that that's not true anymore? And then you can go from there. But mostly, especially when you get to the university levels, they're getting more and more closed. That's what I mean by a divergent. People who suddenly realize, hey, wait a minute, I need to be able to defend my faith. I'm starting to want more of this. But at the university level especially, I think it's getting harder. We used to have, I knew a whole bunch of Christian professors who would welcome creation speakers on campus. I had one here at Augustana College. Other schools, Iowa State had a strong Christian believer in creation in the engineering department. And as they retire, their replacements are all evolution believers. So it's getting harder. That's why you got to teach your children. Get to all the kids' resources you can get.
Right.
Our group, Quad City Creation, if you send me an email, you can answer questions. You can still remember to send me an email. If you're interested in buying resources, we have that. We can send you an electronic invoice and mail them to you. I'll send you the price list. There's my email contact. You can see that, I believe. I'm sharing that.
Yep.
But there's a lot of good stuff out there. We need to get the info out, because if the average person doesn't do it, there aren't enough creation speakers in the entire country to do this. And just by asking good questions, learn to ask questions. Is that 98% number real? Did you ever hear of a tectonic fossil? Turns out it's not a transitional form. Can I tell you about it? Are you interested? I have 4 of my talks, not this one since it's pretty new, but 4 of my talks on DVDs, and you can get all 4 DVDs for $30. And just send me the email and I can send you what they are, and you can decide if you really want them. I can send you the info. I know Twin City has also materials online as well as their own bookstore. But we need to work together more and more.
Right.
We are going to have a Zoom conference on October 3rd. I probably should have had a slide for that. And you'll hear more about that from Twin City Creation, but Twin City and Creation Summit, another group I belong to, are sponsoring that, a Saturday conference, and the main speaker will be Dr. John Sanford, a geneticist from Cornell, who wrote Genetic Entropy, and then also Dr. Danny Faulkner, who was an astronomer, professor in South Carolina for, I guess, almost 28 years.
Can I
Learn
speak?
your resources.
Oh,
Yeah. Another question?
yes. What percentage of Christian schools actually believe the Bible? Do you believe? Because I was surprised about a year ago. A Christian school, a pastor that is the head of a church in Brooklyn Park. I was surprised to hear him say that. Science has proved that man was not created 6,000 years ago. He was created millions of years ago. And all God did was breathe life into a Neanderthal. So I'm just curious, what percentage of Christian schools actually believe the Bible in the United States?
I assume you're talking about Christian colleges and universities.
No. Christian high schools, Christian private schools.
Well, most Christian high schools that I'm aware of are run by the Lutheran Church Missouri Synod. The Missouri Synod is pretty strong on creation.
No, I know a lot that are independent, though. I don't know how many are in here in the Twin Cities, but
yeah, I don't know about high schools around the country. I couldn't answer that very well. I know that there are some. I get invited to some once in a while, but not many.
I was just shocked when this pastor stood up at the end of his sermon and said that, because they have a K through 12 school there.
Yeah.
And but But yeah, I mean, what happens when you only believe part of the Bible? You know, you don't believe Genesis, but you believe the part about giving. You believe this part. You know, that's
Well,
really bad. Yeah.
what's your name?
Oh, it's Brad Hoop. I have a science training, and I visited the Creation Science Museum. I was surprised when I was there, some of the preeminent theologians in America that, you know, have even like one of Billy Graham's contemporaries back in the early 50s. Some, you know, some of these people have strayed from believing the Bible's account. It shocked.
Oh, yeah.
Yeah.
Templeton was a evangelist, a colleague of Billy Graham. They worked together, and then Templeton decided that he needed to further his education, and he went to Harvard and got taught evolution, and then he realized evolution is, if it's true, is just a story of death, disease, and dying is the main character of the plot. And he said God wouldn't use that, so there is no God. Maybe he wrote a book, Farewell to God.
And you have even pastors that, you know, they have been the science Bible dichotomy. They have come because they don't have any scientific training. They don't have the discernment to discern. So they believe science. They believe secular science over the Bible.
Well, that's the issue, is that they
Right,
think the
correct.
secular scientists have figured it out and they're telling us the truth, and they are sincere people. They really do believe it, and they're smart. But they're also blinded. The Bible in 2 Corinthians also talks about, you know, there's a darkness that's coming over their eyes and they cannot see. And so we can show them facts, and many of them will never change their hearts and minds, but they'll just say, well, more research is needed. You'd
Well,
be
this
surprised
pastor,
how strong faith can be in the faith of an evolutionist.
This pastor actually said, he said, it just breaks his heart when he hears Christians saying that they believe the Bible's account.
Yeah,
But they're,
it
you
breaks
know,
his
so,
heart because he thinks he's being scientific.
right.
And therefore, if you are in public and you say you believe the Bible and you can't defend it, you make the rest of the Christians look stupid. That's what he's thinking.
Right, and this is someplace where actually God actually spoke to the pastor to start a Christian school. So yeah, I mean, and they don't even realize they're disbelieving the Bible. And when you start doing that, you now you're picking and choosing what you believe from the Bible.
Well, yeah, and I have a whole talk on this, or many of us do, on the need for this kind of information. What
Yeah,
happens?
I mean, that's the whole issue.
What he has to realize is that he is sowing the seeds of kids walking away. That's
Right.
why 2/3 to 80% of Christians who grow up in good churches, as they are taught that evolution is science, and if it's even encouraged by the pastor, they figure, well, the church has no answers. Why should I believe Matthew, Mark, Luke, and John if I cannot believe Genesis chapters 1 through 11?
Correct. Yeah, there was somebody that spoke at our chapter in the Twin Cities, I think, last April about this very topic. That was his whole emphasis. He ministered to that, you know, the high school crowd, why people were leaving the church, the high school kids, because, yeah, I mean, that's the whole crux of why we're meeting tonight. If you can't believe the Genesis 1 through 10 or whatever, then what can you believe? You know, that's the whole thing. So.
Well,
Okay.
the good news is we've got good answers. We've got to teach our kids, including the high schoolers. And, you know, I don't know if you ever see that pastor very often. All I can do is drip on them and say, hey, you know, the latest science says that we're not that similar to chimps after all.
Right.
Yeah, no, I've tried to send him some other, somebody had actually spoken from our group in the Twin Cities at his church. So she was astounded when I told her what he had said, because they had her speak at the church. But I've left brochures there, and I've sent tweets over there trying to invite them, you know, to our meetings and things like that. Maybe they'll come. And maybe
Okay, that's all you can do. But of course, obviously, the first thing to do is pray.
Yep. Oh, yep. You've got that. You've got that. Thank you. Very good talk tonight.
Yes.
Any other questions about DNA?
Helmut, this is Pat Hurd. I was wondering, are you aware of like any studies? Are they studying like mutations and whether there's like identifying beneficial mutations and harmful mutations, and whether the supposed beneficial ones fixed or stick, something like that?
Yeah. They've been studying mutations ever since we learned about them pretty heavily. And they're really looking hard to find good beneficial mutations. And every once in a while they have a candidate, but it doesn't work out. The most common one that's brought up is sickle cell anemia. That's if you have a mutation and you need it to be, both sides of a chromosome pair have to have it to get the sickness, but it's a very bad disease. And so I've had a young Christian doctor who worked in Africa as a Christian medical missionary say, well, that's a good mutation because she's had a lot of patients who had malaria, and those that had sickle cell anemia didn't get malaria. And the thing is, though, sickle cell anemia is a disease. It's actually, like all mutations, it's a loss of function. You've lost something. So there's a problem with your hemoglobin with sickle cell anemia. Instead of having nice round blood cells, or the corpuscles, they're in the sickle shape. And that's the problem. They don't carry the oxygen as well. And you die from it eventually. The thing is, malaria, which is a parasite that attacks blood corpuscles, is not interested in the sickle shape. So instead of dying quickly from malaria, they die slowly, or their children will die slowly from sickle cell anemia. And they say, well, see, God must have sent that mutation. Well, if there was no malaria, would it be a good mutation? And the answer to that is no. I mean, it's not good. And it's not, see, they're trying to find mutations that can actually add information.
Right.
If that's what you got to do, you've got to make it usable information, then it's got to become widespread. And that was the point of Dr. Sanford's study. In the simulation, he was generous in saying, okay, let's allow 2% of the mutations or 1% of the mutations to be good. And we know even evolutionists will admit that it's probably less, way less than 1%.
Right.
Because they haven't found any, but they just want some to be there. So he said, okay, we'll give you a couple percent. And sickle cell anemia is not a good mutation. It's a loss of information. Yeah, it'll help you fight off malaria, but you're still going to die. And sickle cell, if you really get it, is painful.
Mm.
So yeah, they're studying it. They're trying it. That's their faith. I've had atheist philosophy professors, everything from the origin of life to good mutations. I tell them, the science isn't there. It doesn't back up your story. And he says, they're working on it. And I said, that's good. It's always good to do science. No problem with that. But what you've done is make that a statement of faith. It's not a statement of science. You have faith that they'll solve it. Today, you have no facts. And that's what a faith-based system is. You don't have to have a deity to have faith. You have faith that scientists will solve it, but right now, it's a question of faith, not of science.
Helmut,
Yes.
this is Janine Frichet.
Hi, Janine.
Hi. I read recently, it may have been in Darwin Devolves or somewhere else, but the point the book was making, I believe, is that now since we can actually see genetically mutations, that the idea, neo-Darwinism, that genetic mutations can add up to an evolved being, that basically neo-Darwinianism is falsified. Can you speak to that?
You're talking about Michael Behe's book, Darwin Devolves?
Well, I read a lot of books, and I did read that one. I don't know if it was that or something else.
But basically, you're getting at the point that can mutations create new and usable information, right?
Right.
They can't. In its simplest form, There are a lot of reasons to have a mutation. Just whatever the reason, even just plain normal reproduction, mutations are copying mistakes. And so, when I try to teach people, can you write multiple books that are readable and convey information from copying mistakes when you start with just one page of paper?
That's good.
The bacteria, a simple cell that they say we evolved from, and they have no idea where it came from. They just said, okay, we'll go with it. It's there. It has maybe, what is it? I'm thinking, go by memory, 30,000 base pairs in its little bit of DNA. The human has 3 billion base pairs of information, and it has to all be usable. And we know that now. There is no such thing as junk DNA. So it's like putting a one-page document on a copying machine, make a copy. Hey, it's a pretty good copy. Take that copy at the bottom, put it on top, and make a copy of the copy. And then take that copy, second generation, and put a fourth generation, now put that on top, and you get a copy of the copy of the copy. And you just keep doing that. And you do that 10 times, 10th generation copy, you see the T turns like into an I. But maybe an M turns into an R and an N. Oh, now I got 2 letters, so there was 1. See, that's how evolution works. And by the 1000th copy, all of a sudden I'm getting 2 or 3 pages when I hit the machine. Because I need to have more and more of those letters conveying information. So the idea of going from 30,000 base pairs, or whatever number it is, up to 3 billion base pairs of information means that I'm adding information along the way. And that copies not just have a little mistake, but I'm adding reams of information. Information is the key that also blows away information. I mean, the idea of evolution. You just ask a simple question. If mutations are basically, and even geneticists they call them genetic disasters. They call them problems. That's why scientists are identifying thousands of genetic diseases. Birth defects are increasing because of mutations. You're telling me that's the last hope? That's how I went from a simple cell to a human being through copying mistakes? And yeah, they say, well, they're not totally random because environment chooses the good ones. Still got to have a lot of good ones to choose. And you don't have that.
Thank you.
Oh, I'm sorry. I have another question now, Brad. What about, I heard like fingerprints for the police. When they say they have a match, they have 5 out of 13 matched. And that's supposed to be 94% reliable. But I've heard many lawyers say that their client didn't do it, but they said they had a match. And so I believe a lot of times they don't have a match. What about DNA? When the police get some blood or a hair or some saliva or some semen, are they always reliable, do you think? Or do you think blood is more reliable than semen or what?
If you can get enough of to get a good chunk of DNA, obviously they're not going to, I don't know, it's getting so much faster now. But when they started using DNA, it took a while and they couldn't use all the DNA, but you can get a pretty good match for whose DNA that was. Much better than fingerprints.
How reliable? I believe fingerprints are not as reliable as we're led to believe. And as a matter of fact, I've heard many court experts are not really experts. The tire track matched, the tooth bite matched. I heard a lot of these things are just hoax.
Yeah.
Really not very.
I can't really speak to that.
How reliable do you think the DNA is? If they get some
if
blood,
they get enough DNA, I think that can be pretty reliable. I do. It's from everything I've read. I'm no expert, but that's
like how reliable? 98%, 97%?
Oh, much more than 98. I mean, fingerprints were probably in the 90s. But when you talk about statistics and the chances to get really good matches of some of these, when you're talking about just a few thousand base pairs in a row, that's starting to look pretty good. Okay. It's just how good is the science? I mean, I think they're over 99% if they get enough DNA.
If they match blood, should they match it to blood, or can you use saliva to match blood? Or
it doesn't matter as long as you can isolate the DNA.
Okay, so you could take tissue from the mucosa of the mouth and match it to blood or semen or something. You still think it's
Every cell of your body has a full set of DNA.
wow. And it certainly is used to exonerate people. It didn't match. That's probably even more reliable, I would think.
I would think.
So, okay, thank you.
DNA is information, and most of us have our DNA is very similar, but there's enough places, and I think they've learned where to look, that they can find the differences between your DNA and my DNA, even though a lot of it is still going to be the same because we're human.
Do you know, or are you assuming?
Well, I know we're human, and I know they're using it. That's
No. I mean, are you familiar with the techniques they use in forensics?
I don't know the exact DNA. They use a dye where you get those barcodes of DNA, and they look at that.
Okay.
That I can't tell you. I'm assuming that's what it is, but I don't know that for sure.
Okay.
Just from
'Cause I don't
everything
think-
I've read and assume the DNA match is far better than fingerprints. I think you're right about that.
Yeah, I know. I've heard enough cases from lawyers that I know that fingerprints are not as reliable as they want us to believe. You know, they want us to believe that they've convicted the right person all the time, but they really haven't many times. So, but
Yeah, that's
okay, so, yeah,
another issue.
yeah, okay, good. No, I meant based on fingerprints, but DNA sounds like you think it's more trustworthy.
I'm pretty confident it's much more reliable. Still not 100% because you'd have to do a lot of work to check it all.
Like here, I heard that one time that somebody had supposedly raped somebody, and so they followed the guy around and they got his spoon. They watched him eat in a restaurant and they got his spoon, and they got him because of that. Now, do you think that is reliable? The guy leaves, he leaves his spoon. Now the cop goes over and he has a warrant from the judge to go and pick up the spoon.
You know, I have not studied that particular type of thing, but I do know this. If they can get it fast enough and get a piece of saliva out of there, and there is a cell with DNA, if that's done fast enough, they can do a pretty good job, from what I understand.
How fast?
You got to remember that's why they got to do it quickly and watch them, because DNA, once the cell dies, the DNA deteriorates quickly.
So he has to clear. He has to pay his bill, clear the restaurant, then they have to grab it. They have to have the equipment right there. So how long would that be, do you think?
That I don't know. I just know that relatively, from other studies, DNA decays.
How long would it take for the cell to die on the spoon?
Yeah, I don't know.
Okay. Okay, thank you.
It's an interesting question.
Yeah.
The DNA, the key to DNA is it is information. All of our information is just a little bit different and way different from monkeys. There's no chance that a chimp-like creature evolved into humans.
Yeah, I got that. Thank you.
I want to thank all of you. You've been patient, and we've tried to make this Zoom and recording as good as possible.